And I think a good point to start is acknowledging and appreciating that most of the farming done in sub-Saharan Africa is done by smallholder farmers. And these farmers farm in 0.5 acres to about 2 acres maximum pieces of land. And these pieces of land have high levels of infestation of pests and diseases. They also have very high levels or very low levels of soil fertility. And any small thing that changes these dynamics, for example, the climate change or new pest invasions, exposes millions of farmers to hunger and starvation. And so a technology that is going to elevate or increase farm productivity is a welcome development. So today I'll be talking about genome editing because I think and I believe it has power to increase productivity of agriculture in Africa. To just demonstrate the kind of food that we grow, how many varieties, we grow a lot of bananas, we grow a lot of cassava, we do maize, we do sorghum. So these form part of our staple diets. The question is, how can we be able to increase the productivity of this? We know that there is maize lethal necrosis that limits production of maize. We know that there is banana diseases and viruses that limit production of bananas. And then we have this weed that is very beautiful that limits production of all cereal crops, maize, millet, and sorghum. So how can we use genome editing, or how can we demonstrate here that genome editing can be used and applied to improve productivity in Africa? Today I want us to see how genome editing can be able to change that variety of sorghum that is very high yielding to also have an imparted characteristic or a trait of disease resistance. And the technology that I'll talk about is part of a suite of genome editing technologies called CRISPR-Cas9. I have 2 varieties of sorghum. I want to move the sequence on the lower panel to the sequence on the upper panel. And I said you can use that, you can do that using breeding technologies. And an example for this case is genome editing is the CRISPR-Cas9 system. So you bring in the sequence that is going to search and help you find a region that you want to alter in the DNA, and that is Cas9 associated with the search sequence CRISPR. And what Cas9 is going to do is going to cleave that DNA, and then you have the option of replacing that sequence with an altered sequence to replace the one that was there before. And this process is called homologous end-joining. And what you end up with is a resistant variety that still retains the variety characteristics that were high-yielding. You also have the option of cutting and replacing that with just joining the 2 sequences together. And this process is called non-homologous end joining. So what I just described is a way that you can be able to search a sequence into the genome, and then you can replace it with a new sequence, or you can get rid of that sequence. The sequence that we are removing here is one that we think is adverse to the crop.